Robert Wynn, he's now at the University of Chicago. He was working on junction plakoglobin. More specifically, he was working on this gene in relationship to lung cancer. That what he's found was that in advanced lung cancers, cancers that spread or went metastatic, this protein basically disappeared in most of those tumors. The problem is, and he knew this was probably important, right? Again, we're associating something. Metastasis, the absence of this protein. These two might be connected processes. So he came to me, he's like, hey, I think this gene is really cool. I don't know what the heck it does. He's like, can you help? I was like, yes, I can totally help. Again, it's finding the buddies. So junction plakoglobin, and you can see it basically hangs out on the cell adhesion molecules. Basically, it likes to hang out in desmosomes. And what it does is it basically kind of coordinates the attachment of these cells, or that's what they think it does. So that when you go metastatic, you have to let go of your neighbor. So when you lose this protein, then that cancer gets much, much worse. Okay. So the way I did it is I'm just going to use, Sir Francis Galton's linear regression, right? So we can look at this. This is a positive correlation. So given here is a data set. These are all, the individual lines are different samples or cell lines. The height of the bar would tell you how high the expression of that particular gene is in that sample. So the higher the bar, the higher the expression, the lower the bar, the lower expression. Look at these two genes here. These are two separate genes. What do you notice about them? They look the same? Same pattern over all those samples? Heck yeah, totally. What do you think about that? What does that mean? What do you think? Just coincidence? Anybody? Working together maybe? Could be in the same pathway, right? Why would they all basically fluctuate the same way, right? Maybe they're super friends, right? They're all working together.
League of Justice.
I can always tell how old my crowd is by the reaction. Like the older crowd will laugh their butt off. I'm sure all you are like, why is there a blue monkey there? This is what we used to watch on Saturday mornings. And then, yeah, you look at Robin here. I wouldn't let him near my kids. Like, he's not even wearing pants, so. But again, we're looking for things that are working together, things that have similar combinations that, yeah, they're probably working together. Now look at this one. Two genes again. Here's a similar gene. But isn't the pattern's kind of off, right? When this gene in these samples, when this gene is high, it's low in this one. And the reverse, right? It's high here, it tends to be low here. Maybe they're working against each other, Legion of Doom kind of stuff. Again, you can always tell the age of the group. Yeah, these were real villains. So that's what I'm trying to do, right? I'm trying to get a bunch of friends of JUP, junction plakoglobin, and I'm trying to find its enemies. If I find its friends and enemies, I can figure out what the heck it's doing. To do this, here's the problem with doing correlations, is it's very powerful. You can find associations all over the place, but there's a lot of false positives. So here is kind of the wrinkle in my technique is that I don't just use one data set. I use two data sets, and all the false positives, when I find the middle of it, seem to fall out. So I use, since I'm working on lung cancer, I use two large lung cancer data sets. One is in Dallas, Texas. This was UT Southwestern. They have 76 lung cancer cell lines. They use AFI to do it, basically probing 45,000 different transcripts. There's another completely independent group, the Brodus Institute, or Novartis, the Broad Institute in Massachusetts. They did this cancer cell line encyclopedia. They did 166 lung cancer cell lines, lots of different lung cancer cells, 55 different transcripts that they look at. And again, we're using the same technology, but different chips, but it still works. And if we actually compare the different cell lines in these two different groups, there's 42 common cell lines but there are some differences. Okay? So in order for this to work, junction plakoglobin expression has to vary across all these samples. And sure enough, when you actually look, so this is in the larger data set, when we look, you can see this is a log 2 scale. There's quite a bit of difference in the level of expression here. This is going to give me a good pattern to kind of follow and try to find something that looks like it. Okay, so all I did was just basically do a Pearson correlation test in both those data sets, correlating junction expression with anything that's either positively or negatively correlated with greater than an absolute value of 0.5, an R value. When I do that, I end up with 324 that are correlated JUP, 252 at the UT Southwestern, and 152 that were common to both data sets. This is how I get rid of all those false positives, right? Once I find the middle, I get rid of this, and I only look at this. The odds of all these, me getting this, I haven't done the statistics, I should have percent on this. This is infinitesimal, just based on random chance, that I would get all of these common based on just, looking at 40,000 things here and 50,000 things here. Now what I'm going to do is I'm going to take you through these 152 genes. And by the way, so what I'm going to tell you about now represents about 0.6% of all human genes. This is what I found just doing this simple statistic test. Oh. So here are the top 10 transcripts correlated to JUP. And what we find, and what's interesting, and again, these are the two different lung cancer data sets, is I'm getting the same types of genes in the top 10. And if I don't get the exact same gene, I'm getting an isoform. This is telling me that basically, this is working. That I basically did the same statistical test in two different groups from across the country, and I'm coming up with the same genes. That means that this network is somehow, it's real. It's there. If two groups are seeing this in the transcriptome and I'm seeing it, then this is giving me hope. So this is kind of and we're going to talk about this top one here in a minute, SPINT1. And again, this was the most highly correlated gene to JUP in both data sets. So just to kind of, I'm going to talk about pathways, but I kind of have to talk about the statistics behind how do we know if a pathway is playing a role in a system, right? And it's a very simple test. And basically what I'm going to do is I'm going to give you the cliff notes of all pathway analysis. And this is basically the core of all bioinformatics, is that if I have a pathway here, right? This is a pathway known to be important in, certain types of lung cancer, right? And there's 40 members of this. And I have my 152 genes that are correlated to JUP. What makes this all work is that these 40 members are in a limited number, that there's only a certain amount of human genes in the human genome, right? 25,000 genes. What are the odds if I draw 152? So here's what you can do is you can pretend these 25,000 genes are basically a bunch of green balls, and these 40 are a bunch of pink balls, right? You have 40 pink balls and 25,000 green balls. Giving 152 draws, what are the odds that I draw one of those? What do you think? Not unusual? Probably not, right? But now what if I get 3 of those pink balls in my 152? Now it's starting to get unusual. What happens now if I draw 7? The odds of that happening by random chance are incredibly small. Given this result, I would say that this pathway is probably playing a role in my 152 genes. Again, these pathways we create ourselves. This can be any biological unit. I have all these things associated with a particular biological process. What are the odds that I would get members of that group in my little gene list? That's how pathway analysis works across the board. And we can do that, right? And I can test. So if I think that this gene is playing a role in lung cancer, I should get some diseases or functions that are overrepresented that represent that. And sure enough, when you look at this, lots of cancer comes up, right? And these are in these 152 genes. Again, I told you it was important in metastatic tumors. Again, here is invasion of tumor cell lines. 17 genes out of my 152 are associated with this. My collaborator was very excited to look at that, and he basically drilled into these 17, kind of, what is this? What are these 17 doing? Also, some of the software I use, I wish I could show you today, but based on the level, the direction of the expression of these genes, we can make confidence calls on whether we think this process is more active and more inhibited. And I have other videos to do that. So when we look at the most inhibited processes, when JUP is up, you inhibit metastasis of tumor cell lines. Again, this is further evidence that basically backing up my collaborator that this gene somehow prevents late-stage lung cancer. And then most activated, adhesion of tumor cell lines. Totally makes sense, right? What I'm showing you here is we're taking pathways. I just told you that, I kind of poo-pooed on pathways. But you can flavor them. And what I'm showing you is that we look at all these different types of cancers. It's not pelvic cancer. And we don't see lung here, but I see cancer here and I say, I'm on the right area. And just because no one has researched this gene in lung cancer doesn't mean it isn't there. And in fact, this is a whole new, this gene actually works in lots of different cancers. So now we can bring up the, particular pathway so we can look at cell adhesion more closely. So anything that was colored red was positively correlated to JUP. That was in my 152 genes. Here is Sertoli cell junction signaling. Does anybody know what a Sertoli cell is? Yeah? No? It's basically it helps form the sperm. And that's another problem with pathways, as the Sertoli cell junction signaling will come up when I'm working with a collaborator, and he'll look at me like, I'm not working on sperm. I'm like, I know, it's okay. It's all about cell adhesion. And again, we can look at tight junction signaling. So again, here are all those things in my 152. They're lighting up. This makes sense, right? This is what this gene is doing, and these are the buddies that it's using to do it. Okay. But the real power of all this, correlating gene expression, is finding these networks. So basically, has anybody here heard of ingenuity pathway analysis? Anybody use it? You will. It's a good program. Basically, what this program will do is it'll take my 152 genes and it'll build networks up to, it'll do it as high as I want it. But I think I put the limit here at 35. What it'll do is it'll basically group genes based on any connection, either direct, which would be represented with a solid line, or indirect connections, which would be the dashed. What this program will do is it'll say, what are the odds of me getting all these biological connections based in the literature due to random chance? The odds, so this is my top network coming from genes that were correlated to my JUP. The odds of getting this is 1 times 10 to the negative 70th. That's not random. Basically, when you see that, what we have done is we have, based on doing correlations in 2 different data sets, we now have a network that has evidence of being connected in the scientific literature. And here's the cool part, is that junction plakoglobin here, when you actually look in the literature, the only connection it's known to have is with this gene here, PKP3. Basically, what I have done is I have expanded its network, and now we're looking at all of this, because all of this stuff is probably related to junction plakoglobin. Again, I didn't have to rely on experts, 20-year-old data. I basically, found this on my own. And again, green would be, negative correlation, red would be increased correlation. And I want you to notice this one here too, is SPINT. That now I have a blueprint of what JUP is doing. Now I can attack it and I can look to manipulate this system if I want. right? Because now I know it's true signaling network. And we can overlay certain pathways, right? So if I overlay different things on these genes, what biological groups they're in, a lot in epithelial cancer, neoplasia cancer, that would be the formation of cancer. And again, EMT transition, so this is important in metastasis of the lung tumors. Again, this all makes sense, right? But we're gonna talk about SPINT1, and that's the thing, is you can't validate everything. So basically what I do is I create these networks and I go to my collaborator and I go, this is what I think is really happening. And they have to test it. And he did. So what I'm going to do is I'm going to talk about SPINT1. It's a serine protease inhibitor. It's known to inhibit proteases PRSS8 and ST14, which were also shown to be correlated to JUP. It's a hepatocyte growth factor activator inhibitor. So it's inhibiting growth, which would make sense as a tumor suppressor. And this is what I love, and this always happens, is even if it's not known in lung cancer, it's known to suppress metastatic pulmonary colonization from pancreatic carcinoma cells. So again, this gives us this hint, this connection to the lung cancer metastasis. And again, there's no prior knowledge in the literature of this protein ever acting with JUP. So my collaborator tested this. He looked at a bunch of lung cancer cell lines, and what he noticed is that if there was little to no JUP in that cell line, you also had little to no SPINT. So again, it's that correlation, right? CO2, heat. Got JUP, got SPINT. No JUP, no SPINT. Oh, yeah, forgot I did that. Here's what he also found, is that when you overexpress JUP here in this cell line, SPINT1 went up, right? This cell line has a functional copy of p53, and I'll show you why that's important. So in this cell line, he tried to overexpress JUP, but nothing really happened with the SPINT. Once he puts back in this wild-type p53, now you have expression of SPINT. So what we found was that there's also a mediator somehow in this, p53. That this one has a functional copy of p53, this one doesn't. When you put p53 in, then SPINT1 goes up. And as we were doing this always happens to me. Like, you're doing like a research project, somebody else comes up, they show that JUP is known to interact with p53, but it also regulates the expression of another gene, which actually was also correlated to JUP. So again, this has given us information that it's validating what we found, right? And we can explore this further. And here's what's cool is that some of these, these cell lines have lost JUP, but when you put SPINT1 in here, which actually might act as a mediator for JUP, you have reduced colony numbers. You have less proliferation, right? In both cell lines, regardless of whether they had a wild-type copy of p53 or not. Again, this is another way. Now that we know the network that JUP is in, now we can use SPINT1 maybe to manipulate that, to basically reinstall maybe that anti-tumor effects in these cells. And of course, you would, based on our ideas that this is involved in metastasis, you would hope to see a less migration of these cells. So what they do is they'll grow these cells on plates, and then they'll basically just scratch a little line or basically just wipe the cells out of the middle, and then they'll basically time to see how long it takes to get back together. And so what we can do is, so basically they did that in both these cell lines, except in this one they put in SPINT1. When you do that, when you don't have SPINT1, these cells grow back together in 12 hours. Here, we've put the SPINT1 back into the cell line, and now, even after 24 hours, they still haven't grown back together. Oh, thank you. Appreciate that. I forgot that was dark. So now we have a mechanism, right? And we published this in JBC. You have alpha-catenin, or JUP, somehow induces p53, which induces our SPINT1, which is also known as HAL1, which blocks this growth factor, which then basically prevents cell proliferation and migration and invasion. Or at least that's part of it for sure. Okay, how much time? Okay, I'm going to give you one more example of this. So this was my work in lung cancer. And again, we published this. He now has a mechanism. Now he is basically looking at this as a potential therapy for advanced lung cancer metastasis.